Hello everyone, I'm Sana Musu. Today we are going to talk about editing humanity with Bupe. So hello Bupe,
Hello.
what do you want to say about it
Uh,
firstly?
Basically, what we're talking about today is how editing humanity has now an impact on nowadays. So we'll go through what CRISPR is, we'll go through how it works, and then we'll finalize by looking at the response of the society to this latest form of editing humanity technology.
Okay, how does engineering and medicine cause, from your point of view, from an engineering point of view, how can you explain the impact of engineering on medicine?
Basically, the practice of medicine nowadays hasn't been really an independent fraternity. And what do I mean by this? The department of engineering and medicine, they can coexist, because engineers also help medical doctors reach their wanted standards by actually providing the required equipment for their patients. As the saying goes, behind every medical advance there lies a great engineer. So we can also see in what has happened, in what goes on, like the latest development of an electric nose, and trying to use an electric nose to find cancerous cells and everything like that. So there is a contribution of effort by engineers to the medical fraternity. So these two departments, not only these two departments, but I think nowadays most of the departments are kind of coexisting, because they're no longer independent, they're kind of intertwined. So basically that's how medicine and engineering can coexist.
So, I should say that you are study with CRISPR-Cas, right?
Yeah.
And what is CRISPR-Cas, and what is the long term of it?
Breaking down the acronym CRISPR, the C actually means clustered, the R means regular, and then the I means interspaced, the S means short, the P means palindromic, the R means repeats. So basically, CRISPR is a gene editing technology that enables efficient and unprecedented genetic modification of different organisms and human tissues.
So, from what you're saying, CRISPR-Cas was created, rather designed by doctors or scientists per se in the laboratory. So, was it designed by the scientist, or kindly explain how it works,
So CRISPR actually wasn't designed by scientists. Lack of a better term, it was borrowed from microbes like bacteria. Bacteria use CRISPR as a defense mechanisms. As a defense mechanism, what happens is when they're under attack from a virus, the virus deposits its DNA into the bacterium cells, and as we both know, DNA is basically the instructions of life that contains 4 different chemical units, which are read like a code by the cell. So when the virus invades the cells of a bacterium, they multiply and they burst out of the increase in number and burst out of a bacterium cells in pursuit for other bacterium. So what happens is with CRISPR-Cas, the bacterium is able to defend itself. It gives the ability to defend itself. The Cas part of CRISPR is an enzyme that is normally known as the nuclease. This enzyme is like a molecular scissors. What happens is when a virus deposits its DNA into the cells of a bacterium, the CRISPR enzyme, the Cas enzyme, it cuts or snaps the DNA into two, which means that the virus has been deactivated, and then it gets the foreign DNA and puts it in a special in a certain place of a bacterium genome, and it kind of repeats this kind of creating like a hit list of different DNAs, and then it after later on, after putting it in in a specific genome area, it
Mhm.
copies a sequence into a molecule known as the RNA guide, which also has a Cas enzyme. And this RNA guide is simply, it targets, it's more like a target weapon. It targets specific DNA. So it's like you have a variety of different types of DNA with specified duties. So after scientists understood how it works in bacterium, they decided to copy or adapt to get it from bacteriums and use it in humans and different parts of in the different organisms. So what they do is they get the they get the enzyme, and then you feed it with a specific you feed it with your own RNA guide, which will match the gene that we want to edit. And then when it's put in a cell, it goes direct to the gene that is supposed to be edited, and then it snips it off. But also scientists have improved the entire mechanism, in the sense that they can also replace a cut gene with a whole well gene. So basically,
Mhm.
CRISPR is being used to replace genomes and genes and other tissues in other tissues. Yeah.
Okay, for understanding clearly, what is the purpose of CRISPR-Cas and what are the applications?
Basically, the purpose of CRISPR-Cas, it's used to edit individual genes. It's what happens is that when they used to edit individual genes, they try to see what happens if they switch off these genes. So, for example, in what they're trying to do in human tissues is to see how they can remove the HIV affected tissues, infected
Mhm.
genes from a human, and then also um, to engineer the new genes that can defeat cancerous cells. And
Mhm.
they're also trying to kill antibiotic resistant bacteriums, and they're also trying to use it in plants to kill the weed. And it has a variety of applications in different species. Yeah.
So, why is CRISPR-Cas? Is there any other companies? Are you alone with the industry?
Because quite, it's not the only editing technology that has been developed. There are other gene editing technologies that have been developed. And why CRISPR-Cas? It's because it provides unprecedented functions. It's has like you can use it to cut precise genes. You can also use it to change completely the gene, and basically it has a quicker and cheaper way of doing these things. So
Mhm.
it is widely accessible, unlike other gene editing technologies.
So, I think this is very interesting topic. CRISPR-Cas edit humanity very cheaply and very quickly, right?
Yes.
So, does this mean that we are at the verge of making designer babies?
That's a very really interesting question. It's actually been a topic of discussion for the past years, and just to answer your question, it will take some years, perhaps decades, in order for scientists to use CRISPR-Sky for designer babies. What I mean is, we can look from previous research, like this April, scientists in China tried to edit the an embryo of a human, but unfortunately, it didn't develop to terms. What I mean is, it didn't develop fully. So we can see that there is a lacking in the use of CRISPR-Cas when it comes to editing babies to when it comes to the use of CRISPR-Cas in designer babies. Yeah.
So, if that is the case, what is the response from the society? And I mean, what are the ethical concerns?
The ethical concerns by the society can be categorized into two. There is a practical one and the philosophical one. When we're talking about the practical concerns, as much as CRISPR-Cas is very useful in cutting genomes, it might miss a gene and cut a wrong gene. If we're in a lab, this might be okay, but when it happens in a human, this might create grave danger because it might disturb the mechanism of a human being. So these are the practical concerns that have been raised by people, and truly it is, unless a person is really sick or you're trying to edit genes that are really bad or that are infected so badly, so you can try to edit that at your own risk. So the risk is taken by a person, but the concerns of the people are that CRISPR hasn't been fully developed to a point where it is exactly used for cutting a specific gene, because it can miss and cut a wrong gene. And talking of philosophical concern, quite a number of countries, about 40 or so, have banned the use of CRISPR, and some are still debating. A few other countries are still debating whether to allow the research of CRISPR take place in a specific country. So what people are now concerned with is, as much as it might be used to cure cancer, HIV, and a variety of disease, it's also giving power to doctors to act God. So that has been a concern by human beings, and to cross this Rubicon is actually there will be no return, and it's actually why the people are really concerned when it comes to the philosophical part, because it is allowing doctors or scientists to act God in the sense that they can create whatever kind of species they want to. So basically, those are the two major concerns of on CRISPR.
So thanks for the explanation of CRISPR-Cas and editing humanity. Today we talk with Bupe with editing humanity. Thanks for listening.
Thank you very much.